I work in the cell culture facility at Western Carolina University, and in our lab, we grow human cells to do research with. The cells that we grow are called human embryonic kidney 293 cells, or HEK 293 cells. And this green picture to the left with all the little dots, those are what our cells look like underneath the microscope. We grow our cells because we're interested in understanding how cells respond to stress. We know that when cells are stressed, they undergo a process called autophagy. Autophagy is like a recycling process. When autophagy starts, cells wall off parts of themselves that they don't want or need, and they break them down into smaller pieces that they can use for other things. Autophagy has been implicated in a wide variety of diseases, such as arthritis, Alzheimer's disease, and even certain types of cancers. So it's really important that we understand autophagy and study it. Our lab is interested in understanding how the application of certain biomaterials can actually make cells do autophagy. The biomaterial that I work with is called keratin, and it's a protein, and the version of it that we use is derived from human hair. Keratin has been used in cell research for many years, primarily because researchers have assumed that keratin is inert, meaning that it does not change anything in our cells. However, my research questions this assumption because we actually think that keratin makes our cells do autophagy. When we give keratin to cells that are stressed, those cells undergo autophagy to a greater degree than if they had been stressed and not given keratin at all. So how can we tell if keratin makes autophagy happen? Well, we track a protein in our cells called LC31. When autophagy starts, LC31 turns into LC32, and we can visualize this change using a technique called Western blotting. And so in the bottom left-hand corner, this is an example of a Western blot. And in that blue box, all those little smudges are the LC31 and LC32 proteins changing based on the materials that we provide to our cells. Western blotting is a really helpful tool, but it's really expensive and it's difficult for undergraduate workers to perform. And so what I'm trying to do is to develop a method that does the same thing as Western blotting, but is cheaper and easier to perform. And so in this picture to the right of the Western blot, this is an example of in-gel fluorescence. In the blue box, those are our LC3 proteins. This method is going to be really helpful because it's going to allow us to demonstrate to other researchers that keratin is making our cells do autophagy. And maybe keratin isn't inert at all like we thought it has been. And if that's the case, then we need to rethink the way that we use these materials in our cell research. Because if keratin changes autophagy, then that changes the way that we think about cell behavior in our research.